Once again, welcome to this webinar. I am Meera Kumar, the application scientist at Bellbrook Labs. For those of you who are not familiar with Bellbrook Labs, we are a small biotech company based in Madison. We have been a very reliable reagent supplier for more than a decade and take a lot of pride in our products, and also the fact that we like and we are helping people like you do drug discovery. So let's get on with the webinar today. Transcreener assays relies upon highly selective and high affinity monoclonal antibody that was raised in mouse against GDP that enables selective and sensitive GDP detection in the presence of excess GTP. Now these antibodies are unique in that they are able to differentiate between nucleotides on the basis of very subtle structural differences. For example, in GDP, the antibody can bind to GDP as opposed to 2 phosphates as opposed to 3 phosphates. So that makes it very unique. Direct detection of nucleotides, the only direct detection method available for GDP, and this method is based upon interaction of two reagents. One is the antibody, the monoclonal antibody that's against GDP, and the tracer. The tracer is nothing but GDP that's conjugated to Alexa 633 or other fluorophores in case of FI and TR-FRET assays. So in a coupled assays schematic shown here, what happens is you convert the nucleotide into using different coupling enzymes into a product that uses a reporter enzyme into converting it into generating a signal. So each coupling or each reporter enzyme is a potential target for the compounds being screened, which therefore increases the risk of false positives. Not so in the transcreener assay because it's direct detection and has an extremely simple protocol. So as GDP is formed from your GTPase reaction, the GDP directly goes and binds to the antibody, and this ease of use also makes it possible that you can run this assay in the real time in kinetic mode, and there are no intermediate steps or no coupling enzymes involved. So there are many other assays in the market, and I'll briefly touch upon them. One is Promega recently launched what's called as the GTPase Glow, and as I said, it's a coupled assay, but this is even more convoluted approach to detect GDP. In this method, they use enzyme nucleotide diphosphate kinase shown here that GTP from the GTPase reaction and converts that into ATP, which is then used by luciferase to detect it as light formed or light produced. Now, so this is basically a depletion assay, and as is true for any kind of depletion assays, you will need to deplete 40% or more to see a good signal, and at that condition, you're not actually following Michaelis-Menten parameters anymore. You're not at initial velocity. So that's one of the drawbacks of depletion assay, and once again, it uses a lot of coupling enzymes. So it's multiple additions and it cannot be run in real time. Also, DiscoverX has ADP Hunter assay that they say can also be used for GDP detection. Once again, has let's see, about 1, 2, 3 enzymes here that converts it into a colorful compound that can be colorimetrically measured. So those are the drawbacks of coupled assays. So why go for transcreener? Because that's the only direct assay available for detection, and just because we understand that there could be customer preference and there could be instrumentation barriers. Some instruments are only capable of measuring one detection format. We just introduced it in 3 different flavors: the FP, the FI, and the TR-FRET. The FP has been the workhorse and the first assay that was launched at Bellbrook. So this assay principle basically shows that the antibody is in its native conformation. There is no modification made to the antibody, and the tracer is an Alexa 633 633 tracer, a far red tracer. And once again, just to iterate that we chose far red tracer to minimize compound interference. GDP formed in the reaction, the antibody is bound to this tracer. That's a bulky molecule now, and it spins relatively very slowly. The rotational mobility is very, very slow because it's a bulky molecule. Now, as the GTPase gets activated and GDP is formed, the GDP has a higher affinity for the antibodies, therefore goes and binds to the antibody, and the tracer is released. Now, this tracer, just GDP that's bound to a fluorophore, is a tiny molecule, and it tumbles really fast and therefore changes the plane of polarization. So as you can see, this is a negative assay, fluorescent polarization. So as GDPs increase, your polarization decreases. So it's a negative assay. But the positive is that the antibody is in its native conformation, unmodified, and therefore the reagents are really, really rugged. You can, we have done experiments where we have left the detection mixture at 37 degrees for 3 weeks, and yet it performed as good as fresh reagents taken out of -80. So, we don't encourage that our customers do so, but just to prove our point that how our reagents are extremely stable, we have done such experiments. Now, fluorescent intensities, we take the exact same monoclonal antibody, but now we attach it to a quencher molecule. And as you can see, in the absence of the product from the GTPase reaction, the quencher is quenching the fluorescence from the tracer molecule. So you start with relatively low or very basal level of fluorescence, but as the reaction progresses and as you make more GDP, what happens is the quencher is, or the tracer is displaced out of the antibody and gets unquenched in the process, and you start measuring the fluorescence. So it's a positive assay in the sense that as the product GDP is formed, you see an increase in fluorescence. The TR-FRET version is, again, you take the same antibody and you now attach it to terbium chelate, and there is a FRET in the absence of GDP, and the FRET is removed as GDP is formed. So the principle of the assay is very, very similar between the three formats. As the tracer is released, you either measure the change in fluorescence by FRET or fluorescent polarization, or just measure the relative fluorescence. So the principles are very similar. It's just that the antibody and in the TR-FRET formats. Now, how does it impact like a customer? The assays have slight differences. The graphs below show that we have to either optimize the antibody and the tracer mixture. Now, the Transcreener assay itself requires that you detect GDP in the presence of excess of GTP. Now, the concentration of the antibody in your reaction, therefore, determines the total assay window and the sensitivity of the assay. So, when you get started, you will probably have to do an antibody titration or a tracer titration, as in the TR-FRET assay, to get the best assay window. But to make it very simple for our customers, what we have done is streamlined the whole process. So in our technical manual, we give these graphs that shows that there is a linear correlation between the concentration of GTP, the initial concentration of GTP that your enzyme would use, and the amount of antibody that you would require, or the tracer. So there is a linear correlation, and there is an equation. So you just plug and play. You put in the amount of GTP in micromolar that you would use, and it would tell you the amount of antibody or the tracer that you need in your reaction. You could also do, I mean, we have had people who like to do it all by themselves. Therefore, they run a binding curve, or what we call an antibody titration or a tracer titration, and determine the EC85 concentrations, which is the most optimal concentration to give a good assay window and a great sensitivity. Or else you can just plot it in this equation and get started from there. The only thing that you need to pay attention is that sometimes if your buffer has some more additives that, we optimized all this in a very simplified buffer system, but you might have other components in your and just to ensure that they don't interfere with the detection reagents, it's always good to do a titration as well. Moving on to the next slide, this is just a competition experiment where we have titrated GTP and GDP, and this graph compares the FP, FI, and the TR-FRET formats to show that the antibody is very selective for GDP over the GTP diphosphate over the triphosphate, and you see that the selectivity is almost like 138 to 140-fold selectivity for all three formats. The assay also has great sensitivity and robustness. The three examples, like the examples shown here, are standard curves. And the way, I would take a minute here to go over how we recommend running a standard curve. And the idea of a standard curve is to mimic enzyme reaction, a GTPase reaction in this case. So let's take an example of 10 micromolar GTP being converted into GDP. Now, at 0% conversion, we have 10 micromolar GTP plus 0 micromolar GDP, no GDP, only the triphosphate at that point. At 50% conversion, we'll have 5 micromolar GTP plus 5 micromolar GDP, and at 100% conversion, we have it all converted into GDP. So there is 10 micromolar GDP with no GTP. So the idea is to mimic an enzyme reaction where you decrease the substrate and you increase the product at the same time. So we keep the ratios of the nucleotide constant. So shown here are examples of 1 micromolar, 10 micromolar, and 100 micromolar GTP to GDP curves in all three formats. And down below is a table that shows the Z prime. And as you can see, if 10% conversion, which is kind of considered the standard for initial velocity conditions, you see that we get really robust Z prime greater than 0.9 in most of the cases. So the assay is really, really sensitive. So an ideal assay is one that, as I mentioned, has great, robust, and signal stability. So here we show FP, FI, and TR-FRET assays, and basically I'll walk you through what it means. The first graph here talks about deck stability, and the deck stability is when so this is an example of a 10 micromolar GTP being converted to GDP. So deck stability is when you leave your detection mixture, say you prepare your detection mixture ahead of time, you add the tracer and the antibody and then leave it at 4 degrees for a day before. So you either prepare it an hour before or you prepare it like 24 hours ahead of time. It shows that the deck stability of the reagents for all three formats is extremely good. You get almost the same readout. The TR-FRET is slightly poorer than FP and FI, as you would have noted, but the FP and FI have excellent deck and plate stability. So the bottom panel of the graphs show plate stability, and this would be extremely important for people running high throughput screening assays. So you run your assay, say it takes you a long time to set up Thursday evening at 4 p.m. and you got to rush home, you can come back the next day and read the plate, provided you seal the plate to minimize evaporation, so you do not have evaporation. So if you seal the plate really well, you can come back the next day and read your plate, and it would show excellent plate stability as seen in FP and FI, and a little bit in TR-FRET. So for TR-FRET, we say 8 hours is what we kind of say in our technical manual. So as long as it's 8 hours, it's really good, but you can still get by with a 24-hour plate and deck stability if need be. So at this point, I have talked enough about the Transcreener technology, and I hope I have you convinced that the direct detection is a really good system to go with, and the reagents are very stable, and you have a choice of 3 different detection formats based on your preference, instrumentation, and also based on sensitivity. I mean, I did show that the FI probably did not have as rugged of a signal and plate stability as FP and FI. So you kind of, I hope I have you convinced that there are 3 formats that you could choose from. Now moving on to the main topic of the day, GTPases. So just a little bit of introduction before I show you some data. GTPases are again a family of hydrolase enzymes, and they bind to GTP, and the hydrolysis takes place in the conserved G domain shown in this graphic here, and it converts it into GDP. Now, all regulatory GTPases have a very common mechanism that helps them to switch the transduction on and off. Okay, the toggling of the switch is performed when GTP is bound. It's in an inactive form to the GDP-bound form by hydrolysis of GTP through intrinsic GTPase activity. Okay, that's the switch. Now, this whole cycle is called the GTPase cycle. Now, most of the monomeric GTPases have really very slow intrinsic GTPase hydrolysis rate. The G-alpha proteins, the heterotrimeric G-alpha proteins, have slightly better intrinsic GTPase rates, but the monomeric proteins that most of us are interested in have very, very slow intrinsic rates. Now, this is an example of the monomeric GTPases, and again, I'm showing the data here in FP, all three formats, and there are 4 enzymes that were produced at Bellbrook Labs. We expressed it and purified it at Bellbrook Labs, and shows that they're very similar in their performance. This is just an enzyme titration showing the activity, the change in polarization, or the change in fluorescence, or the change in Fret ratios for the 4 enzymes. Now, this is, I just threw in this slide just to show the comparison between the TR-FRET, FP, and FI assays. Now, sometimes when there are 3 different assays, I always get the question, which one do we choose from? Are they all similar? And this just shows that the EC50 that we get between the 3 or all the targets, very similar, and for all purposes of rank ordering, even with IC50 values, we get very data from all the 3 formats, so they're very, very similar. It's just personal preferences and instrumentation that you have to pay, that makes the decision between the 3 formats. Now, I'm going to talk about GEF proteins a little bit here. Now, the GDP, like I said, the GDP dissociation from the GTPases is very, very slow. That is often the rate limiting step. The GDP dissociation rate is often the rate limiting step. So the binding of the GEFs to the GTPase substrates catalyzes the dissociation of GDP, then it allows the GTP molecule to bind in its place. So GEFs, the protein's function is to promote the dissociation of GDP, and after GDP has dissociated from the GTPase, GTP binds in its place as P to GDP is 10 to 1, so the GTP immediately goes and binds in its place. And results in the release of the GEF protein. Okay, so this GEF both destabilizes the GTPase interaction with GDP and also stabilizes the nucleotide-free GTPase until a GTP molecule could bind to it. So we'll get to GAPs a little later in the presentation, but this is just the slides. The following slides would just talk about the action of GEFs. And recently there has been a considerable interest in the GEF proteins because they have been identified in a lot of cancers and as an oncogenic targets. So there is interest, there is an emerging interest in this new class of proteins. So what we have done is, by adding the GEF protein, you increase the dissociation rate of GDP, and when you add your transcreener detection reagents here, you can measure the amount of GDP produced using, I mean, shown here is an example of FP, but you can do it with either FI or the TR-FRET assay. So how do you go about measuring the activity of GEF proteins? The first and foremost would be to pick the concentration of your intrinsic of your GTPase. Now, CDC42 and ROA don't have, you can still detect the amount of intrinsic GTPase activity of these two monomeric GTPases. For some proteins that I will show later, it's almost undetectable, like ARF protein. You cannot detect the intrinsic GTPase activity. It's really negligible, so you have to add a gap or a gap to see it. But if your GTPase is something where you can detect the intrinsic GTPase activity by incubating it for a longer time or increasing the temperature, the first step would be to pick a concentration of your enzyme that has produced 20% of the signal or has very, very minimal intrinsic GTPase activity. So for the CDC42 and ROA here, I pick somewhere around 30 nanomolars, 40 nanomolar, and 80 nanomolar concentration, which gives me only around 50 MP shift, really low. So that's the first step. And then you titrate your GEF protein, just GEF by itself, to see that there is nothing funky going about happening with the GEF protein alone. And then on top of CDC42 and ROA, the concentrations that you pick, to see if you see a GEF activity, do you see an enhancement? So that's how you determine the concentration of the GEF protein that you want to use and the GTPase that you want to use in your assay when you're developing an assay. So this was a recent publication that came out of collaboration from Bellbrook Labs and the Lancano Institute. And the first two graphs showing CDC42 and ROA was generated. This data was generated at Bellbrook Labs, and the ROB data was generated at the Lancano Institute. But the point here that I want to show is when you have CDC42 by itself, the blue trace that you see here, you see very little intrinsic GTPase activity over time. But when you add the GEF protein, the graph in the red trace, you see that there is a significant increase in the polarization shift that you see. And this, it's very similar for ROA and ROBs. You see that when you add your GEF protein, you see that there is an increase in the signal that you get. Now, our assays, the Transcreener assays, are by nature, so I should back up a little bit. I'm getting ahead of myself here. All competitive binding assays by nature are nonlinear. This is not just true for Transcreener. Any competitive binding assay is going to be nonlinear. So you would see a response such as this when you plot the raw data in the y-axis. Now, and that is why we recommend that you run a standard curve. It's not mandatory, but if you want to see what does 80 MP shift, or what does 60 MP shift, how much GDP does it correspond to, you will have to run a standard curve under similar conditions, and you can extrapolate that data using graph data that you see here into the product form GDP. And as you see, that is linear. So so to see the product formed, you will have to do that kind of data analysis. You will have to run the standard curve and convert the raw data into the product form. Now, if you look at the graphs below in the bottom panel, you would see that the product formed is completely linear, and you can see the effect of GTPase alone and GTPase plus the GEF protein. You see an enhancement. So we use that, we use these numbers to calculate the rates, and we express them as turnover numbers, the number of GDP molecules that are formed per minute when you add the, by themselves or by when you add the GEF protein, and the ratio of these values is what we call enhancement, or the GEF effects, the enhancement due to this GEF protein. So as you can see that in all three examples that we used, CDC42, ROA, and ROB, we do see some kind of GEF enhancement due to the DBS protein that we used. This is another example of a P-Rex protein. P-Rex is RAG-specific GEF. It is phosphatidyl inositol triphosphate dependent RAG GEF protein. Now, there have been recent publications that shows that Prex is has been highly upregulated in prostate cancer, and hence there has been, as I said, there has been an emerging interest in Prex as an oncogenic target. And we show here as an example the effect of Prex upon RAG protein here, and as you see, this is Prex by itself, or sorry, RAG by itself, and here is the GEF activity, shown as raw data here as polarization change, but the increase in polarization that you see by addition of different concentrations of the Prex protein. Now, once again, as I mentioned, the data here would be nonlinear. That's completely normal because that's the nature of any competitive binding assay. And if you want to see linear graphs, all you have to do is run a standard curve and convert these data into the product form, and you see these linear curves. And once again, you can calculate the turnover numbers and the GEF effect. And as you would expect, at higher concentrations of Prex, you would see as compared to lower concentration. With this Prex protein, we also ran an orthogonal pool screening. Now, this is again a collaboration with Lancano Institute, and it's slightly different from the typical screening campaigns that most of you would have undertaken. Here, what happens is each well contains 10 compounds from a compound library, and this compound library itself is unique because it has many core scaffolds which are not found anywhere else. Now, the compound is represented in 2 wells, okay? So there are 10 compounds per well, and that one compound appears somewhere else in the whole plate, somewhere else. To be considered as a hit, the compound must show reactivity in both wells. And so that's kind of it. Why is this better than the typical screen? Because it sort of costs less, because you can do in almost like 6 or 7 plates, you can screen a lot many compounds, and it conserves the use on reagents also. So that's kind of some of the advantages of using an orthogonal pool screening campaign. Now, we took this Prex RAC GEF activity through the 6400 compound screen. So as the 6400 compounds were could be fit in 4 pre-dispensed plates. So once you request for a plate, LCGC, Lancano Institute would pre-dispense them in the kind of plates that you want and send them to you, and you just run your assay. You just add your substrates and start the reaction. And for GTPase, it's fairly simple, right? You just add your GTP and initiate your reaction plus or minus the GEF protein, and then read the plate after an hour, and then we send the data back to LCGC for deconvolution, because they are the ones who know which compound is there. And then you can get the for follow-up screen or for dose response curves like we show here, you can get the compound from them plated, and we can do that. Dose response curves too, and that's what we did here at Bellbrook Lab. And here are some of the screen statistics for the Prex RAC screen. The Z prime was 0.8, which was very good, and that's the control for the Transcreener assay. The Z factor of the entire screen was 0.6, and we got with plus or minus 3 standard deviations considered as the hits, we got 21 compounds. And when followed upon, there were only a couple of them that came up as hits. And unfortunately, I won't be able to share the structure, but it kind of gives you an idea of the hit rate that we go for. So I'm going to shift gears now a little bit and talk about gap proteins. Now, gaps are GTPase activating proteins, and as we talked about earlier, the GTPase, the intrinsic GTPase of activity of most monomeric proteins is very, very slow, and the gap proteins act as activating proteins, and it increases that GTPase activity. So you can increase this K-cat of the GTPase by adding the gap protein. Now, I'll be talking also about RGS proteins in today's webinar, and they also act as gap proteins. They increase the intrinsic GTPase activity. So once you increase that, now you can measure the dissociation of GDP using the Transcreener assay. I do have to say that we haven't done any experiments where we have both gap and the GEF added together. I mean, that would be really cool. I mean, we're planning on doing some studies that way, like you have your GTPase, then you add your gap to increase the intrinsic GTPase rate, and then add a GEF also to increase the dissociation rate, and then measure those turnovers and see how those rates increases. And we're planning to do such studies in the future, but we haven't done those yet. I'm going to first talk about ARF gap, and this this data was generated at UNC, and the publication came out in 2011. So this talks about the ADP ribosylation factors called ARF gaps, and unlike CDC42 or the other monomeric GTPases that I showed, the intrinsic GTPase activity of ARF proteins are extremely slow. It's almost undetectable, and can only be detected if you add an ARF gap and measure, the GDP production from there. So the data that I would show you would be, would cover that in just a little bit. So this is an example of gap, and I'll run you through this experimental setup. So in the x-axis is just the time. So as I said, with our Transcreener assay, you can monitor the production of GDP over time, I mean in real time. So the blue circle here shows no ARF gap, so the intrinsic activity of ARF protein is almost undetectable. But as you increase the concentration of ARF gap, you see, I mean, just like the GEF protein, that it was a dissociation of GDP, but here it's the activation of GTPase, but you do see a decrease in polarization with the FP assay, and once converted into the product formed GTP hydrolyzed or GDP, you see the linear kind of graphs that I mentioned before. This is an example of pilot screen run with the ARF gap protein, and of a low-pack library of almost 1,200 compounds here. The graph B here shows that the correlation coefficient for two screens that were done was about 0.98, so it was really good. And this just shows the scatter plot of the changes with ARF1 and ARF gap 1 of the compound library. So I'm going to, the last thing that I'm going to talk about today is about the RGS protein. And so as I have been mentioning, the problem with developing an assay for the heterotrimeric proteins is the GTPase of GTPase activity of the heterotrimeric proteins is a little fast, but the GDP dissociation rate is slow. Okay. Now, so we need to slow down the GTPase a little bit and increase the GDP dissociation faster, and that's when we can study the changes. That's when you can study the effect of the RGS protein or the gap protein. So you need to we need to do that. So a possible solution is adding RGS. When you add RGS, which acts as a gap for G-alpha, it causes an increase, or it makes it less slow, I should say, the GTPase activity, and makes the GDP dissociation. So what we did here is we made some changes to the G-alpha protein here. We kind of did some mutations in the G-alpha protein, and to make it better suited to study the RGS. So there were two mutations. One was the arginine to methionine mutation, which causes a 100-fold reduction in the GTPase activity, and the other was from as alanine to serine mutation, which caused an increase in the dissociation rate of GDP. So these two mutations were performed in G alpha, and we have a patent filed for this, the mutations that we did in the G alpha protein. And we kind of do it as a system to study RGS protein. Here in this particular graph, what we are showing is the gap activity of RGS4. So in x-axis is the time that it takes, and you see the GTPase activity of G alpha itself has been considerably slowed down because of the mutation. But then when you add the RGS protein, you see a gap enhancement. And as before, you can convert the data into product formed and calculate the gap enhancement. So around a 9-fold enhancement was seen by adding the RGS4 protein. Now, this is again a 960 formed with this RGS compound. This was Gen Plus library, I believe, Gen Plus library with 960 compounds. The Z prime for the assay was 0.83, and we got around 17 hits for this, and 10 of them happened to be GTPase inhibitors. So the RGS4 inhibitors were just like about 0.7%. That was the hit. So the point that I want to convey is using this system, using RGS protein and the G alpha protein that we have, double mutated G alpha protein, we can run screen, and we have done some screening services for customers too in the past using this system. And we've done some study here showing that this double mutant strategy. So we have a panel of RGSs that we have tested, and this has, and we did this double mutation with the other G alpha proteins, and we show that most of them did work very well for us in our hands, and we have a patent filed for this system for us. So that brings me to the end of our webinar today. So just to summarize, Transcreener assay is the only HTS method available that allows direct detection of GDP found by GTPases. And we have this assay available in FP, FI, and TR-FRET format. The assays are very robust and reliable, gives great signal stability, and has been validated in several high throughput screening campaigns in the past. It has withstood the test of time for more than a decade. Many people have tried out this assay, and we can use Transcreener GDP assays to detect the modulators of GTPases for gaps and GEF activity, and we are currently pursuing this area with interest, especially the GEF protein P-Rex is something we have been pursuing now. So with that, I'm just showing the 3 products that are associated with the GDP assay. We have fluorescent polarization, intensity, and TR-FRET assay. The catalog numbers are here. If contact us, and these are just list of all the enzymes that were validated in the Transcreener using the Transcreener assay. If not just for GTPases, if any of the other families is of interest to you, let us know, and we can share some additional information with you. And with that, I thank you for joining me, and I will take questions. So the first question that I have is, could you please comment on the affinity KD value of the Transcreener antibody towards GDP and labeled GDP? Yes, we have found it to be close to between 2 and 4 nanomolar. That's the affinity that we have measured using assay format in-house. And there is a question number 2. Hold on just a second. Could you comment on the way the fluorophore is coupled to GDP, guanine part or the diphosphate part? Unfortunately, that's proprietary information, and I would not be able to disclose that information. We have IP filed on it, so sorry about that. I won't be able to disclose that information. The third question that I have is, when you're using the FI assay in a gap-assisted hydrolysis assay with GTP-loaded RAS, is the assay measuring free GDP, or can it actually attach to the nucleotide that's still attached to the protein? I would say the assay actually measures the free GDP and not to the nucleotide that's attached to the protein. It's very specific to just yeah, it would be measuring the free GDP there. I think that's all the questions that I have, or maybe that. That's all the questions that I have for now. But as I mentioned in the beginning of the presentation, we have a recording, and we will send you all the copy of the slides so you can go through that. And should you have any questions at all, like after the fact, feel free to contact us, send me an email, or just shoot an email to our info box or to me, and I would be more than happy to answer them for you. There is one more question. In an assay with GAP, do you always need a GEF to free the GDP? No, not always. It might take a association rate is just slow, but it eventually comes out. So you can monitor it over time, and we have done all the studies that we did here with that ARF gap, especially there was no GEF involved in that study. So just adding the gap, the GDP will dissociate. It might take longer, but so typically these gap and GEF assays, it takes about 2 to 3 hours to to get a good signal, unless you add a higher concentration of protein, which we don't want to use. So if you follow it for like around 3 hours, you should be able to measure that without a GEF. But what I was trying to mention in my talk was it would be interesting to see the effect of gap plus GEF if the turnover is like really fast. We haven't done those kind of studies yet, and we are actually doing that right now. So I'll keep you up. I have another question. Can you read the signal online by combining the detection reagents into the reaction mixture? Absolutely. So so the stop mixture that we say is nothing but EDTA containing buffer. So it just chelates off the magnesium and stops the reaction. So most of the GTPase work that I showed today were done in real time, because it takes you one set of experiment to set, and you can take multiple readings over time. So the way you do it is you start your enzyme reaction, you have your GTPase, then you add your GTP plus your tracer and antibody, everything at once, mix the plate and read every 15 minutes. The antibody takes about 8 to 10 minutes to equilibrate, and by that I mean, as the GDP takes some time to find the antibody and bind to it, right? That takes about 8 to 10 minutes, but once after that, it's like real time. So we take the reading 15 minutes, and every 15 minutes after that for however longer you want to go, like 3 hours, 4 hours. And if you say after 4 hours you think you've got enough signal and you want to stop the reaction, you can add EDTA at that point and stop the reaction, and then come back the next day. The signal would still be very stable. So that's the beauty of the direct assay. You can start the assay as real time, and you can stop it at whatever time you want, and then continue reading the plate just to make sure that it's good. Or you can even read the plate, like I have done experiments where in the day, like by 9, I have the reaction going, and I take up to 4 or 5 hours of read through the day and come back the next day and read it too. And it's nice to see the turnover numbers, because some of the GTPases, if you're just measuring the GTPase activity of a protein, it's excruciatingly slow, and you might want to come, sometimes it takes like almost 8-9 hours to read to get a good signal. So it's nice to keep it going, and you could do that. I have another question. Can you detect GDP concentration in vivo? Unfortunately, not with our assay, and it's because inside a cell there is very high concentrations of GTP, UDP, GD. There are so many nucleotides there that could interfere with the assay, and the presence of nucleases. So our assay is not designed for using with cells. It's kind of a purely biochemical assay which requires recombinant enzyme. I have another question from Andreas. With 2 to 4 nanomolar Kd, have you tested GDP and ADP? Yes, unfortunately, yes. This antibody also binds to ADP. It binds to ADP also. So you cannot do them both in the same well. You cannot detect both GDP and ADP. But we kind of feel like for GTPase, you're adding GTP and you're measuring GDP, so you're just detecting GDP. Unfortunately, you cannot do GDP and ADP in a combined assay. You can, yeah, sorry about that. Yeah, you can't do that. Okay, I think that's all. Or there's one more. Okay, if you set up the assay minus the enzyme, preheat at 37 degrees for more than 15 minutes, take out and add the enzyme, does it still need another 15 minutes to re-equilibrate? Let me read this again. If you set up the assay minus the enzyme, so assuming the tracer, the antibody is in there, would you still need 15 minutes to re-equilibrate? I think so. I would say not 15 minutes. I would say more like, it's between 5 to 10 minutes, actually. That's the real number that we have determined. It's around 5 to 10 minutes, but that's why we don't recommend doing like the burst you can do, like the first 5 minutes would be inaccurate. But there is I have done some experiments, to be honest, I have done some experiments like that, where I run a standard curve side by side, and I read the standard curve also at 0 minute and 2 minutes and 5 minutes, and you can use those numbers to actually determine the GDP, and that's okay. Then it kind of balances it out. So if you had a control, say a standard curve run under exactly similar conditions, and you can go back and convert your raw data into the product formed from 2 minutes and 5 minutes. I have seen that it works out really well. It's okay. But just because if you're publishing that data, your reviewers might come back and say, hey, if the equilibration rate is 10 minutes, then how are you determining that? So that's kind of a little difficult to address, and that's why we say read at 10 minutes and you should be fine. Do you compare FP, FI, and TR-FRET assays with regards to false positive rate of compounds? Yes, we have done that study, and it's very similar because the assay itself, all of them use very similar fluorophores. I mean, they are by themselves a little different. For example, FP is Alexa 633. For FI, we use Alexa 594, and for the TR-FRET, we use Highlight 647. They're all in the far red region, and we have done I believe GenPlus or Lopac, I can't remember quite right. I mean, one of those commercial compound library, not a huge one. I believe it's GenPlus, the 1000 compound library, and the hit rate came to be very similar between the 3 different detection formats. So I would say the false positive rate is very similar. The FI was primarily launched, because it's at least to me, like when my enzyme is forming a product, I would like to see a positive response. So that was something that our customers felt so too. So we launched the FI just to kind of have a positive readout, and also the fact that most academic labs, I mean, at the beginning, like when we started launching the GDP assay, most of the work was done GTPases. It was many academic labs were interested in it, but they had a big hurdle. They didn't have a FP reader or a TR-FRET reader. All they had was a fluorescence reader. So we wanted to launch an FI assay, but they're very, very similar between the three formats. I would say, if at all anything, the FP and FI are more sensitive than TR-FRET. I've always found TR-FRET to be a tad bit lesser sensitive than FP and FI. I think that was the last question. And if you have any other questions, please feel free to email us, and I'll address that via email. And thank you very much once again for joining me today. And you will be hearing back from us with a copy of a link to the recording and also to the slides. Okay, thank you very much.